so we'll go ahead and get started. So again, everyone, my name's Emily. Thank you so much for joining us. First, I do want to thank Dr. Dolan for giving us the opportunity to discuss mass spectrometry today, and also for organizing the event. But before we do begin with the technical part of the workshop, I would like to start with a brief overview of our lab and the services that we provide. And then after that, I'll turn it over to Dr. John Howe for the technical presentation on PTMs, and then we will have a Q&A session. Right, so some of you may already be our clients, but for those that haven't heard of us, we are located in Frederick, Maryland, and we do specialize in protein mass spectrometry. We also offer sequencing services such as whole genome sequencing, direct RNA sequencing, and whole plasmid sequencing. We use Oxford Nanopore technology for these. As well as we do offer traditional Sanger sequencing and can even support microbial identifications and genome typing projects. So as you all know, life sciences research has evolved significantly over the last century to where we are now looking at the molecular level. So with that, we need to analyze the genome, the transcriptome, and the proteome. So at Puchon, we do offer a wide range of proteomic services using mass spectrometry technology. And here's just an example of one of our instruments. We use Easy Spray for the ionization source and Nano C18 columns. So here's an example of the general workflow for our proteomic services. We take care of the sample preparation and the method optimization. We also do the data analysis and can provide raw files if you wish to do additional analysis. And here's a depiction from a technical perspective where you can see the MS1 and MS2 spectra. And we generally use data-dependent acquisition. So you may already be aware of the biological applications of mass spectrometry, but in general, we can identify proteins from any biological sample and detection in at the nanogram level. So for protein profiling, we can identify differentially expressed proteins to discover potential biomarkers or to analyze how different treatments can affect the proteome. We also can identify PTMs, which you'll learn more about here soon. And we also do intact protein analysis for molecular weight determination and other custom analyses. So just a few quick case studies here. This is an example of 24 different samples. They were each subjected to a 110-minute run. Over about 2,000 to 5,000 proteins were identified, and that even includes the target proteins, which in some cases were at extremely low expression levels. So here's an example of a project that was published where they were looking at the interactome of RAD52 protein. So they were comparing RAD52 IP of wild type to knockout and mutant to knockout. And in both cases, over 1,000 proteins were identified. And of those, over 200 were determined to be statistically significant. And you can see that visualized on the volcano plots here. And then for the same project, this visualization is just the classification of those changed proteins. Over 50% were determined to be nucleic acid binding proteins. So this is an example of a quantitative proteomics project. They were comparing two drug treatments on a cancer cell line. So the left heat map shows over 7,000 proteins were identified, and of those, 51 were determined to be significantly changed. And this is the same project, but just visualizing volcano plot for the two different treatments. Now moving on to intact protein analysis, we can analyze monoclonal antibodies. And here you can see the clear glycan profile in the bottom large spectrum. We can also reduce the sample and analyze the molecular weight of the heavy chain and light chain separately. So in this case, you'll see two peaks in the spectrum. Here's the light chain enlarged and the heavy chain enlarged. We can also analyze antibody-drug conjugates to determine the drug-to-antibody ratio. On the right-hand enlarged spectrum, you can see the major forms of the lysine-linked ADCs and the glycan forms detected. And in this particular project, 100% of the drug was conjugated. So as I mentioned, we also provide genomic sequencing services. This graph just shows the genome sizes for a variety of organisms. So you can see how large the genomes truly can be and why we do need modern technology to advance research. So our core technology is nanopore sequencing using the Oxford technology platform. And this image just shows the principles of the technology where a motor protein feeds a single molecule through the nanopore. which then results in electrical signal changes that can be decoded or base called to determine the sequence. And here's just a broader overview of the technology. The advantage of this technology is its long-range capabilities and the ability to sequence RNA directly without the need for PCR. So the applications, here's some for whole genome sequencing can be used for the completion of existing reference genomes, identification of undiscovered genes and associated function, characterization of uncharted regions such as centromeres and telomeres, and identification of genomic variations, your SNVs, SVs, insertions, deletions, CNVs, STRs, and modified bases. For direct RNA sequencing, applications for analyzing full-length transcripts for accurate isoform identification, base modification, the length of poly A tails, and also can rapidly identify and characterize RNA viruses. So here's just an example of the general inputs and outputs for whole genome sequencing and direct RNA. The good thing about this, we know your samples are very precious, so only less than 3 micrograms of sample is actually needed for these projects. Here's an example of a project for whole genome sequencing of a tumor cell line. On the left-hand side, you can see the coverage across each chromosome for an average of 89%. Based on the total reach, the coverage depth was 45x. Over 5 million variants were detected. Of those variants, 909 were classified as clinically significant. And here's an example of a project for direct RNA sequencing of RNA isolated from 293 T cells. On the left-hand side, you can see the classification of the transcripts identified. Over 30,000 were completely matched to the reference. These transcripts sequence corresponded to over 19,000 unique genes with overall transcriptome coverage of about 85%. And for our standard services for whole plasmid sequencing, we do deliver data the next day. You will receive a report with the read summary and a consensus sequence, as well as fully annotated plasmid map.